Welcome everybody online and in person to our last seminar of our spring seminar series. I'm Sarah Batterman. I'm an associate scientist here at the Carey Institute of Ecosystem Studies. I'm delighted to introduce today's speaker, Dr. Xiangtao Xu. Dr. Xu is visiting us from Cornell University, where he's an assistant professor in the Department of Ecology and Evolutionary Biology. Prior to that, he held postdoc positions at Harvard University and University of Notre Dame, and he did his PhD at Princeton University and his undergraduate degree at Peking University. I've known Dr. Xu since we were both PhD students at Princeton, down the hall from each other. He was in geosciences and I was in ecology and evolutionary biology. And what has really impressed me the most about Dr. Xu is his uncanny ability to bring together numerical models and multiple different streams of empirical data, from ecophysiological measurements to forest inventories to flux towers and remote sensing. And he uses these data and these models to conduct experiments that answer questions about vegetation-environment interactions under novel global change, especially in tropical forests. And this ability to combine numerical models and data is a really essential skill and approach to solving and understanding the future of the biosphere under global change. So I'm really looking forward to Dr. Xu's talk. He's going to be talking about predicting tropical forest growth and mortality under global change. So please join me in welcoming Dr. Xu.
All right. Thanks so much. Thanks, Sarah, for the nice introduction. It's great to be here, especially as a Cornell EEB person who has always talk about Carey all the time, and it's good to have an in-person visit finally. And I'm glad I'm the last person in the spring. That means I bring summer to you, hopefully. So I'm going to talk about tropical forest, predicting tropical forest dynamics, and that's kind of been a big focus in my lab in the last few years. And at the end, I'll talk some other more local project that I think would have some relevance to a lot of other project going on at Carey. So here, I'm showing a picture I took in Luquillo, Puerto Rico. It's a atypical tropical forest in some sense, right? So it's very wet, 3,500 millimeter rainfall. So you would expect, very tall canopy, like dipter carved style forest. But the canopy is quite open in that sense, right? It's the top of canopy is probably only like 15 to 20 meter tall. There are a lot of, you see cecropia, palms going on instead of lake successional giant trees. So I'm not going to talk about Luquillo today, but I feel this is a good starting point where it shows what are the key factors that are of concern for tropical forest dynamics in the future. They are recovering from hurricanes, so they're having a big disturbance going on, which is increasingly more frequent to shape tropical forest. They are regrowing under higher CO2 world. We want to know how much that will influence their dynamics. And they also have a compound effect of a more frequent drought in 2015. So all these factors are together to shaping the forest, right? And another thing is regeneration. We're always talking about reforestation in tropical forests. What's the potential of regenerating forest in helping us to mitigate climate change? So all these factors kind of highlight the important environmental stressors or factors we need to concern under climate change. So a little bit background. Forest Ecology 101 for tropical forest is very important in global carbon cycle. This is an old paper back in 2011 about one of the very early synthesis about forest carbon cycling across the world, right? So you can see the tropical intact forest and regrowth forest have a large contribution to the total carbon uptake or sequestration globally, right? Of course, there are deforestation going on, other things. And from a global carbon cycling perspective, they are very important for variability as well, not just the stock, but also the variability. Well, of course, if you look at seasonal, it's mostly temperate. Ecosystem dominate. But if you look at interannual variability, decadal, and then centennial, the tropics are, at least to our best knowledge, they are the majority source of the variability of the carbon cycle. So that means we need to really understand their environmental sensitivity to understand what's going on in the future, right? And this is Institute of Ecosystem Studies. As an ecosystem ecologist, the first thing we do is accounting yearly. So this is some of the accounting of the main carbon fluxes over in the tropical forest globally, right? Based on multiple sources of data in the review paper, it shows like the intact forests and regeneration forests are drawing down 1.4 petagrams of carbon per year and 1.6 petagram of carbon per year. And there is balanced by deforestation. So there's a net source in general based on those data back then. And then 2015 hit with a big ENSO drought, 2015, 2016. And a lot of different data sources suggesting that the tropical forests become a net source of carbon during the drought for various reasons: soil respiration, reduced in productivity, and increased mortality. There are different ecophysiological reasons. So that event brings us this question that really concerns both, the forest ecologists, but also people who worry about carbon and climate changes. How resilient is tropical forest under global change? That kind of overarching question. First, because it has a lot of carbon stored there, and if you're going to lose them, that means additional carbon to the atmosphere. Second, they are providing so much different kind of ecosystem services for carbon cycling, for water cycling, for climate regulation. And if we're going to lose it, that will influence all these parts as well. So basically, in some sense, if I wear my Mahler's hat, we're trying to predict the change of carbon in the future in tropical forest, right? And it has been uncertain for maybe 2 decades or even more. So this is one of the classical paper talking about uncertainty of terrestrial biosphere models into the future, where they are kind of constrained historically because they're all constrained by the same data set. But then they start to diverge in the future, whether it's super resilient and increased carbon sink because of CO2 vaporization effect, or one of the model, I believe, is Trifid or whatever, or Halley model, that have major mortality in the Amazon. So Amazon becomes savanization of Amazon. And there's a huge range. And if we only look at tropics, there's also a wider range depending on which climate model you're using and which vegetation model you're using. So it's highly uncertain. So if it's uncertain, that means we don't have predictive power. That means we don't have enough information to design optimal mitigation or management or plans to protect them. Okay, if we get back to the mass balance question at ecosystem scale, if we want to predict delta C, we want to know how much is coming in and how much is leaking into going out from the system. So it's carbon gain and carbon loss. So there are a few factors that can influence these separate processes. I'm not going down to ecophysiology, but some general factors. A lot of hope is given to higher CO2 can help us, right? Because that can boost up the carbon gain with CO2 vaporization effect. And there's reforestation efforts trying to increase the regeneration forest, protect them to sustain the carbon gain. And from loss side, we talk about heat stress, fire. There are some fire issues, especially in the edge of the forest. There are drought. We're having more and more frequent drought. And there are also storms, which we start to work on more recently. I know, even not here, but even it's very interesting storms and lightning and other things as well. So all these things will influence these two terms. And today, so basically this kind of shaping the research program we have, we want to understand how important each factor are and how to model them so that we can predict the future fates of tropical forests more realistically. Oh, and one thing I'm not going to talk about is deforestation, like the land use change and other things today, but that's super important as well. So the main part of my talk, we'll talk about two recent papers, and the study in preparation is focus on growth and mortality separately. So try to figure out the biggest factors that influence the growth and mortality. So the first one is, will tropical forests grow more under higher CO2? Will higher CO2 help us to combat climate change, right? So it's a collaboration with Professor Peter Zuidema from Wageningen, from Netherlands. And going back to this, the motivation of this study is, if we look at these outliers of the model that predicted increase in tropical carbon stock and increase in carbon sink, those models predict that because they have a super strong CO2 vaporization effect. Productively, MPP goes way up under higher CO2 world. That creates a negative feedback in the system, right? But recently, in the recent years, there's a lot of discussion and uncertainty talking about the magnitude of CO2 vaporization across the world. Like in a few years ago, I think maybe 2 years ago, there's a paper on Science saying recent global decline CO2 vaporization effects on vegetation photosynthesis. That stirs a lot of debates with a lot of responses. You can see a lot of the questions, whether that's true or not. And last year, there was a paper by Trevor Keelan et al. They gather all the data available, try to do emergent constraint on the CO2 vaporization effect on GPP, gross primary productivity. So their analysis showing the average effect is 0.6. That means every percent change in CO2 increase will incur 60% increase in GPP, right? So that sounds like, okay, maybe we will see higher growth rate under higher CO2 world. But let's step back and let's think about the process involved. So if I wear my ecophysiologist hat, what's going on under the hood, and are there things that are missing? So first, the whole CO2 vaporization effect is we are emitting the CO2 by burning the photosynthesis that have done in the past billion years, right? And then we have higher CO2 in the air. Then it makes the increase the gradient of CO2 from the air to the cell, intercellular space of the leaf tissues, makes it easier for CO2 to diffuse into the leaf. So you have a higher CO2 concentration within the leaf. Then biochemically, you will enhance the photosynthesis, right? Based purely on biochemical principles. And then there's more photosynthase will help trees to grow faster. So get bigger trees and stock more carbons on the land that reduce the increase of CO2. And as I shown in the last slide, there are a lot of evidence, although there are debates, but there are a lot of, like from growth chamber to ecosystem level experiment to large scale synthesis, shows consistent evidence that there is strong CO2 vaporization on productivity or photosynthesis. But we're missing one link there is, does increased photosynthesis necessarily translate to more growth or more structural carbon accumulation, right? So there is another process that involved is the carbon use efficiency or biomass production efficiency, whatever you call it. And it has really mixed evidence for that. People are talking about nutrient limitation. People are talking about changes in allocation strategies of plants. So many things going on. It's not, and it's hard to measure, right? It's easier to measure GPP, the fluxes now with all the satellite data across the world, but it's hard to measure tree growth. So the project motivated this. We want to see, okay, there's some natural experiment going on for hundreds of years because we have been pumping CO2 into the atmosphere. So we can look at the tree ring records. For example, this is a large log of Celia tree in the Huai Kai King Wildlife Sanctuary in Thailand. And it's probably over 150 years old, something like that. So think about all the tree ring records that stored information about tree responses to CO2 increase for about 100 ppm or so. Okay, it's different from the phase experiment, maybe smaller magnitude and also more chronic, but can we tell something, get something out from this natural phase experiment? And Peter Zuidima's lab has, like decade ago, has done some empirical analysis. They collected tropical tree rings from 3 continents, both Africa, Amazon, and Southeast Asia. And they try to collect hundreds and thousands of tree cores and try to count, identifying the tree rings. So some tropical species do have tree rings as well. And it's common, like a few, maybe 100 or more species. And you can count the tree rings and try to associate that with tree growth, right? You have the individual level tree growth data. And what they found, they also measured the delta C13 within that rings for selected species. And they can back infer the intercellular CO2 concentration, the CI. Okay. So what they found is there's definitely an increase in the carbon isotope, a change in carbon isotope that can translate into increased intercellular CO2. So because on higher CO2 in the atmosphere, there are higher CO2 in the cell and higher CO2 signals within your tissues, right? And that will translate into increase in water use efficiency, intrinsic water use efficiency, because on the higher CO2, the stomata will close a little bit more, or you have fewer stomata, so you actually lose fewer water per grams of carbon you gained. That's consistent with what we know from gas exchange measurements and the phase analysis. And what's surprising to them and to me as well is when they standardize the tree rings after taking account of ontogenetic changes and other things, there's no trend. And it's not just that site, but also in Congo, but also in Colombia. And it's also, no matter whether you group species together or do it individually, most, if not all of them, have non-significant trend, right? So what does that mean? That means maybe they're not growing more under the historical 150 years CO2 fertilization. Well, and I'm a modeler by training, and so my question is, okay, this is a cool data set to see how well our model is, right? Most of the model are like thinking onward, but what about thinking backward in time? So can model, and I'm here to ask, like, can models calibrate or trained using the contemporary data? That's what all models are doing right now in the last 20 years of data, or last 30 years of data. Can those models trained with contemporary data capture the historical trends in tree rings under long-term gradual CO2 fertilization? If not, we can simplify the growth, the woody growth, as GPP multiplied by woody biomass production efficiency. So how much of that can convert to biomass? If there's a bias, is that because the model is biased in GPP, or is that a model biased in the production efficiency allocation, right? And then can we infer in the constraint growth sensitivity to CO2 and also climate variability from the tree rings? I'm not going to talk too much about that. I'm focusing on the CO2 fertilization story. So to do that, a challenge is how to compare apple to apples or tree ring to tree ring, because most of the models are operated at ecosystem level, right? They don't have tree rings. And tree rings are inherently biased samples. First, you're only looking at survived trees. Second, you're only looking at ring-bearing trees. Third, you're only look at probably have a biased sample size because usually you're targeting big trees that can have more rings, right? So there are a lot of inherent sample biases. So we try to minimize that. So we're taking advantage of our ecosystem demography model, so at 2, and which has individual level processes. So we can actually simulate the growth history at semi-individual level. It's cohort-based, not exactly individual, but we can get more comparable growth record. So we want to do that. So what we end up doing is we develop a digital tree coring algorithm to try to find the surviving trees in the model simulation at the end, and then go backward, try to find their corresponding growth history. And we're finding exactly kind of species and the size distribution to make sure it's comparable. Okay, trying to reduce that sample bias as much as possible. So, and we designed some numerical experiments where we first constrain our model or benchmark our model with contemporary forest structure and seasonality observed there. We conduct two historical simulations where one simulation we have realistic CO2 increase in the past, so there are more CO2 in the atmosphere from 1800 to 2000. The other one, we hold it constant at 285 ppm. So then we can then tease out the other effect. Maybe there are climate effect and other things as well. We want to tease out the CO2 effect. Then in the model, so in the real data, we have real tree rings. And in the model, we can do virtual tree coring, and we got growth history of each individual. So each line is an individual in the model, right? And you will see differences by comparing them. All right. So first, I want to show you the model is well constrained contemporarily. That's what every modelers will claim in the first figure of their paper, saying our model is great, right? So what we're doing is we want to show it's comparable to the observed for structure and fluxes. So the white bars here is the basal area structure as a function of size groups in the Huaycai 50 hectare forest geoplot. And the stem densities here, and this is the GPP. We don't have the on-site GPP. This is the flux tower 100 kilometers away, but kind of representative. All right, and the orange bar is the CO2, the realistic CO2 increase simulation, and the blue bar is the constant CO2, so no CO2 increase. So as is expected, there are more trees in the orange simulation than the green simulation, sorry, and they're more comparable to the observation. There might be some overshoot in some areas, but for example, for big trees, where the CO2 increase simulation are doing way better than the CO2 constant simulation. We're not doing too bad on the stem density. We have underestimated seasonality. The mean GPP looks good, but we're overestimating seasonality, in part because we focus on deciduous species that have ring bearing, and the flux tower footprint is more a mix of evergreen and deciduous. So we're not too worried about that, but the timing and the seasonality looks pretty good. Okay, so the first question is the model consistent with the tree ring data? So we simply look at a trend. So this is the temporal trend of standardized growth for two plant functional types within the model. So one is more softwood that grow fast, and one is more hardwood that grows slower maybe, but more resilient to stress. In both observations, when we look at post-1950, so we focus on post-1950 because we have more reliable climate data, and there's a rapid increase, relatively more rapid increase after 1950. So in 60 years of data, actually in the standardized growth is negative overall. So trees grows less and less in those 60 years. While in our model, we predict so this is softwood, we predict very large positive trend in both simulation because in part they're recovering from some disturbance in the past. So that's why you see in the baseline constant simulation still are positive. But you can see in the CO2, this simulation is higher than that one, right? So there is a positive effect in growth. And for the hardwood one, it's more interesting, right? The model is, the observation is more negative, but the model is non-significant. So it's non-significant. However, if we kind of unpack that into different size classes, because it could be some community cascading effect where if your top of the trees grow better, they shade more of the understory trees. If you group all the trees together, you can have mixed signals. So we try to separate the size classes. If you look at relatively mid-story and smaller trees versus the large trees. So mid-story, like smaller trees, the observation is way negative. So they're growing worse, either because of shading or because they have fewer access to water or nutrient. We don't know. And interestingly, in the model, we do have more negative trend than the baseline. So the baseline has no trend, but in the model, have a little bit negative trend because of that competition effect. So you have more overshading and other things. But if we look at the overstory, which are not shaded, should be free from all these effects, The observation is very positive. And the constant simulation, constant CO2, of course, there's no trend, right? There's no effect, but the increased one has increased as well. So you see, there's a complex story going on. For some species, maybe their model is overestimating. For some species, maybe the model is doing well. And it's also a function maybe of the forest structure. Okay, so we ask for the ones that are potentially biased in growth. Is it because the model is biased in productivity or carbon use efficiency? We don't have historical productivity data, but we do have the interstellar CO2. So the best thing we can do is compare the trend of interstellar CO2 in both the model and the observations. So we found that, as expected, if we have constant CO2 in the atmosphere, there's no trend in CI in the model. There's a little bit negative because of drought and other effect, but very slight. And interestingly, with our CO2 increase, we got really very comparable magnitude of increase in CI. Okay, although it's not CI multiplied by the Rubisco activity will translate into GPP, but the Rubisco, there's some evidence about Rubisco acclimation, but we did that magnitude analysis. It's not enough to cancel that effect of enhanced GPP. So overall, we believe the GPP bias is small because the model can't capture the CI. All right. So the simulated CO2 effect on GPP is likely reasonable. So then we back calculate the woody production efficiency. So basically, we try to say this is the raw production efficiency in the model. So it's about 18 to 20%. That means 20% of your photosynthesis will finally end up as structural, above ground structural carbon. And then if we force the model to match the observed growth pattern, we can see a gradual decline of the biomass production efficiency. So going from about 20% down to about 12 or 13% in the past 60 years or so, which is a function of atmospheric CA. So that means if we really want to force our model to match the observation, a more reasonable way to tune the model is to reduce their carbon use efficiency. And our analysis cannot distinguish which processes drove that change, but some reasonable guess is increased NPP allocation to below ground, either to overcome water stress or nutrient limitation. And interestingly, that sensitivity is very comparable to a very independent phase experiment in Australia in the Uke phase site. They reported similar sensitivity of reduction in WBP comparable to the tree ring data. Okay, so what's the takeaway from this individual site level analysis is the terrestrial biosphere models, which are all built on Farquhar model, are likely able to capture tropical productivity response to CO2. So that's probably okay. In contrast, the models are likely unable to capture tropical growth response to CO2. There are two reasons. One is we need to consider functional diversity. Different species might perform differently, and size, structure matter. And then there is a potential large carbon allocational shift is necessary to explain that response. So there are a lot of hypotheses about progressive nutrient limitation and other things, but again, I feel like we really need more data and theory to understand that. And the ones that I didn't show is we also found for climatic analysis is the productivity efficiency actually increased during hot and dry years to make up for the reduction. So they draw their carbon reserves to increase the production, to maintain production, woody production, although their GPP has reduced. So while in a model, most of the time it's not that case. So we need to consider all these adaptive strategies plant might have. Okay, and then overall there's a lot of untapped information from tree rings for the modeling community with a lot of more and more individual level demography explicit model are being developed. And what's going next? We're trying to work with Peter to expand from one site in Southeast Asia to maybe multiple sites in multiple site there, and also potentially globally. Try to try to understand how tree grows, not just flux, but real tree growth going to respond in the future. And if step back, I think if we think on more ecophysiologically, I think that points us to shift away from resource acquisition, which has been a dominant modeling framework to understand the tropical carbon dynamics, like how efficient you are to doing photosynthesis, but more about storage and allocation. What's the best, how should we consider those processes, right? The turnover of the non-structural carbohydrates, the allocation to respiration, and the fine reproduction, defense, and all the other things. How do we put that into the framework to really help us to do long-term prediction? Okay, so that's my first story about the growth. My second story is talking about the second term, the loss. And this is based on a recent paper on GCB and a larger analysis we're doing right now. So if we're going back to this carbon loss, okay, so these are here again. There are many factors, but we actually don't know the sensitivity for the loss term to a lot of these factors. So the overarching motivation is, if I have to write a model, how can I figure out the equation to use, right? And it's of a real concern as well, like in the past, from the synthesis of 300 plus field plots over the Amazon, people are thinking the mortality is increasing in the Amazon, which is responsible for the declining of net carbon sink, right? So this is a great effort. However, scaling from this 300 plot level mortality to regional scale is really challenging. Think about that. Every plot is at most 1 hectare. It's hard to do repeated census, right, in remote areas like the tropics. So think about to represent 9 million square kilometers using 300 hectare, right? The scaling is a real issue. And because of that data challenge, the environmental drivers of spatial temporal variations remain very unclear. So I would call it a theory-rich but data-limited problem. There are a lot of theories about what are the factors: drought, storm, lightning, temperature, vividity, many papers. But usually they're very limited in data to support the result. So motivated by this, our key questions are, okay, can we get data to do spatial pattern analysis? Is it possible to directly monitor fine-scale mortality from space? Okay. Once we have the data, what are the environmental drivers of spatial temporal variations of tropical forest mortality? And then think about the consequences. Where are the mortality hotspots under climate change? So monitor mortality from space has been a keen interest in the remote sensing community for a while. And there are many tools we can use, but they have their own limitations. So first, more direct measurements of structural biomass is LiDAR. So when you shoot a laser from the sky, you can, based on the waveform bounce back, you can estimate the vertical profile within a 25-meter footprint. And that can directly measure structure or biomass. However, the current spatial coverage and temporal resolution cannot separate growth and mortality at fine scale. So usually people bundle all these individual shots into 1 kilometer or even 2 kilometer and bundle them into net change in biomass, which is the sum of growth and mortality. And in tropical forests, growth is very high. Mortality is also very high. So basically, you only have their difference. It's hard to back in for the individual fluxes. More recently, there's another way using the microwave remote sensing from satellite like SMAP or SMOS. They measure the microwave signal bounced back from the forest, and the microwave signal is very sensitive to water content. So you can use that signal to estimate vegetation optical depth, which is a measure of vegetation water content. So the good thing about these satellites is global coverage, high temporal resolution, daily to weekly. However, they have coarse spatial resolution, like 10 kilometer to 30 kilometer, and they can't separate the water and the biomass dynamics. So you can see a signal in a dip signal. You don't know whether it's their water stress or biomass loss, right? And they can't separate growth and mortality as well, because you still only see net change. And the third route is the traditional optical remote sensing, for satellite like MODIS, Landsat, that people are more familiar with. And the good thing about them is global long-term coverage, fine spatial resolution. So you got the reflectance from multiple bands across the EM wavelengths. However, the challenge is reflectance is not a direct measure of biomass. So we need to figure out how to infer biomass. So our motivation is I really need long-term observation, a fine-scale observation to resolve that. So let's see how much we can do with that. So in our first kind of case study, we focus northwestern Costa Rica. I'm more familiar with the ecosystem there, and also there are tropical deciduous forests, so it has more cloud-free days compared with Amazon, so easier to work with there. So we focus on the 2015-2016 ENSO drought because, so you can see the drought really kicks in 2000. So they start a little bit drought in 2014, then 2015 the peak, and recover in 2016 and 2017. And the good thing is we have like in situ mortality measurements. We do see a big increase in stem mortality rates. So more than doubled from the ground measurements. So we want to see whether satellite data can help to pick up that. And there is a theoretical foundation for our analysis is with the mortality going on, the signal from the EVI or vegetation indices should have increased autocorrelation because you're having a pattern now with decline and recovery, especially for the high frequency variability. So we basically want to test whether after removing seasonality and long-term trend, whether this autocorrelation signal can be used to quantify mortality. So on the right-hand side, that's our results. We show that these are on y-axis the relative forest mortality observed from each plot for 4 years. X-axis is we use Landsat, so we know exactly, we have very comparable footprint of the signal. This is the EVI residual autocorrelation. So there is a very nice nonlinear relationship. If you have high autocorrelation, that means something's going on in the forest. And we compare with traditional metrics like anomaly or trend of the vegetation indices that are used to were used to identify mortality. They're not doing very well. More scattered. But if we put into autocorrelation framework, it actually is much nicer. Okay. So then we applied that masters are trained in this area to try to estimate the regional carbon loss. Again, this is just mortality, not the net change, it's the mortality. And then we found that in the core area, in Guanacaste, in the Santa Rosa Park area, where the epicenter of the drought, we have really big changes in mortality rate during 2015 and 2017, and it bounced back a little bit in 2017. So it varies from like 2% all the way to over 10%, right? And these are regional area, on average, the whole forest region in northwestern Costa Rica. And we estimate the total above ground biomass loss due to drought-induced mortality is about 1.38 tetragrams in northwestern Costa Rica. And we did some additional analysis for the driver, but I'll skip for that, but focus on the ongoing analysis to expand it to the whole Amazon, right? So this is what we found in Costa Rica. And we extend analysis, analyze terabytes, like 40 terabytes of data from 2000 to 2020 over the whole Amazon at 500-meter resolution. We collect all the forest ground data we can find and try to see whether the same pattern follows. And I was worried at the beginning because the scale mismatch, right? You're talking about 500 meter by 500 meter versus 1 hectare, forest plot. But it seems there's still a signal. Very similar and more scattering, right? And when we try to do cross-validation, we found that our estimate is relatively unbiased and with a root mean square error of 2.34 megagrams of per hectare per year. So that's better than nothing. And there's definitely a lot of pattern, especially for big mortality events. So we have more scattering in the small mortality events because there's very little signal in the canopy, right? Okay. So then we go out, try to run this algorithm spatially. So this is the pattern, multi-year average mortality. Again, this is not net change, but mortality. Most of them are balanced by growth rate, right? These are the pattern distribution for the whole Amazon basin that are classified as forests. And we mostly focus on intact forests because our training data set are all in intact forests. In degraded forest, it's more challenging. There are land use change, understory fire. We don't want to deal with that, but we do see a longer tail in the distribution of mortality in the disturbed forest. Okay, and the general pattern, it seemed to make sense. You have higher hotspots in the southern Amazon where there are more fire and the land use change, and there are more mortality along the river where there are more disturbance, the flooding and other things. Okay. And we use the most up-to-date biomass data to calculate biomass turnover time because that's usually the way the big model are thinking about how to model carbon loss is your existing carbon multiplied by a turnover time. Okay, so this is basically mortality divided by standing biomass. And we see some interesting pattern going on. So the median of the carbon above ground carbon resident time from our analysis is 51 years in the Amazon. So about 2% loss per year, which is consistent with field data, a lot of field data. And you see interesting pattern where it's low in the central the river basins, and it's very high in the Guyana Shield in the northeastern part of the basin. And that's the place where you find tall forest, hardwood that are, that have really high biomass. Okay. And then with that data, we can do some spatial analysis. Now we have not just 300 sites, we have the whole basin. So we set out to do some machine learning analysis to try to understand the drivers, the environmental drivers of the biomass turnover rate, turnover type. We tried a few factors, going from this side to this side. This is the drought, maximum cumulative water deficit, long-term average. This is soil fertility, the soil cation, concentration of cation. And higher value means more nutritious in general. But again, a lot of uncertainty in this data. This is the temperature of the warmest quarter to represent the thermal stress. This is the elevation because in part is we don't know what's going on. There might be patterns for lowland forests, more flooding and other things. And then this is CAPE, convective available potential energy. That's an indicator of the intensity of convection and storm and nightly. And interestingly, the most important one, so the parenthesis, the number in the parenthesis is a measure of their importance for spatial pattern. So you can say the most important one is CAPE. So the reason why we see tall forest and low turnover rate in northeastern of the basin, the Guyana Shield, is because there are lower risk to storms and lightning. Okay, and that really explains like 15 years or more. So that the low storm and lightning risk basically say that increased their turnover time by 15 years or more. That's considering the average is 50 years. That's a lot. Okay. The second factor is elevation. It has a weird nonlinear pattern that's hard to really interpret to the first order. But what I'm thinking is this part showing the influence of floodplain forest and the human disturbance. You can see it's in this area along the river basin where you have more flooding and human interventions. And then this flat, and then for the really higher elevation, it's lower. In general, it's a lower turnover rate means higher mortality, right? So maybe that's really with soil texture and other things, soil depths and other things that needs more investigation. Temperature. That's something interesting and consistent with our understanding. It's mostly flat until there's a kind of a tipping point that maybe 27 degrees C, then it starts to decline. So the turnover rate starts to decline. Turnover time. So that means you're under higher mortality risk when you're under the edge of the 27 to 28 degrees C in the temperature. So this is the heat stress. Soil fertility, that's something probably Sarah can explain better than me. It's very interesting. There's no response in general until except for the very, very nutrient poor soil. So that means if you look at the residence time, the nutrient poor soils draw down the residence time, so they're higher chance of mortality and turnover. I don't know whether that's true and what are mechanisms, but this is what showing up in the data. Finally, originally, like, people thought drought would be very important, but it turns out like drought is the last variable. But again, some of this might be correlated with elevation, so statistically it might be hard to separate them. But you can see there's also some kind of tipping point. After a tipping point, you start to have higher mortality and lower turnover time. Okay. So based on these analyses, which we're still refining, we found that we can leverage optical remote sensing to scale ground-based mortality to regional scale. That really help us our analysis. And this creates opportunities to benchmark terrestrial biosphere models. And we found that storm-related disturbance might be a very potent determinant of Amazon forest mortality regimes. And most of the models don't consider that mechanistically. And also, our data can provide novel constraints on mortality sensitivity to storm and drought. And finally, of course, a direct result from this is regions with increasing CAPE. There are regions, because of climate warming, the air has more energy and water sometimes. So there are regions with increasing CAPE. Those might be mortality hotspots under climate change that we need to care about. Okay. Okay, so to wrap up, in those kind of projects, we try to focus on this equation, and we found that the higher CO2 might not be a silver bullet to help us, right? It's questionable how much they can help to boost the carbon gain. Yeah, and then the storms are I wouldn't say neglected, but under considered process that are influencing carbon loss. So in the future, we really need to get these two right in the model to constrain our uncertainty in the future. So that's my main story, and I will take a few minutes to try to introduce my lab and other project going on that might be interesting. So as you can see, like from the projects, I define myself as ecologist and ecophysiologist in silico. So we're trying to develop numerical greenhouses with computers and to evaluate the hypothesis. And also try to basically create some kind of crystal ball to help us to understand the future fates of the biosphere. And to do that, we need to integrate information from ecophysiology, how they work, theoretical ecology, how to scale up, and computational and data science, right? How to ingest all this data. And then we combine like in situ observations, and we're now starting a control experiments in Puerto Rico. We're doing through-fall exclusion, try to understand at the ecosystem level how the vegetation responds. And also leveraging the increasing petabytes of data from multi-platform of remote sensing. And some of the other research direction going on is, that's really going to my heart, my research is to predict a functionally diverse ecosystem in the tropics, right? Especially there are hundreds of species coexisting in 50 hectares. And how can we consider that? What are the leading dimensions of interspecific variation? And how do we consider intraspecific variation? And can we use some eco-evo optimality theory to help to constrain that hyperdimensional space in terms of modeling? And then, as I mentioned, we have an NSF and DOE-funded project to monitor and model ecohydrology of a new through-fall exclusion experiment in Puerto Rico. We're setting up the whole structure over this summer, and with understory roofs to divert the water. And then we're marrying everything we can think of, like sap flow, soil, hydraulic distribution, and other things. So if any of you want to work there, that's a new facility, especially if you're interested in drought. That's a new facility you can consider. And finally, with some money from USDA and New York State, we're developing a local project to use terrestrial laser scanning and to calculate to estimate forest carbon sequestration and the carbon MMRV, all these more applied questions. And also, I'm also interested in the plant structure diversity, how they deploy their leaves and the canopy. And to kind of introduce a little bit more about that is the terrestrial LiDAR basically is shoot millions of lasers per second to reconstruct your 3D surroundings of the forest structure. And then we take that raw data and try to understand data to develop algorithms to segment individual trees. These are all forests near Ithaca. young forest or mature forest, and then we can do it within time. We do digital census, so we can track 3D growth and branch turnover over time. So these are the 2 small trees we scanned in 2 years, and you can already see in the colors means the growth they have done in the last year. So I feel these will really create novel data set for us to understand the ecophysiology and the ecosystem consequences of their allocation strategies. And another kind of project that I was just talking with Winslow is about spongy moss. So in Thompson County, we had like 2-year outbreak in 2011, 21 and 22. And here it sounds like they're 3rd year now. So we're trying to see how can we really monitor and model it from the space. So this one is showing why I want to do that. Is this curve area polygon is what the USDA Forest Service are doing with aerial mapping, aerial sketch mapping. So they have airplane, somebody draw on the on the airplane, try to draw polygons. So this is area they identify as outbreak, and we use 5-scale satellite data to calculate the intensity. So brighter areas with higher intensity at 10-meter resolution. We show that yes, they're right, but there are much more details going on. Very heterogeneous. There are hotspots, there are like a cold spot, and mostly probably because of species differences. And then we can scale it up to a whole Tompkins County. This is Tompkins County in 2021, where we have ground-based measurements, observations of outbreak. So you can see the hotspots in a few areas, right? But there are a lot of areas that are not influenced. Some of them are evergreen. There are species that the moss doesn't like to eat. Maybe there are other reasons. And now we're trying to scale it up to county level for the whole New York State. And this is in 2021. So we have epicenter here, epicenter in the west, and a little bit in the east. And we're now trying to extend it to 2022, 2023, and see whether we can see any hotspots in the east. And with that data, we can do more spatial modeling, saying, okay, what are the drivers? Are they inherently population dynamics, or there are environmental cues we can use to understand when are they going outbreak? Another project maybe relevant to some of the Cary people is we're having a side project for inventory ponds with remote sensing, especially the water bodies below 1 hectare. So here is we're developing some machine learning based algorithm to do high resolution remote sense data to try to find all these little ponds across the space. So this is Hamilton County in the Adirondacks. This is Tompkins County with more agricultural and man-made ponds, very different characteristics. And one thing I want to show is it's very important to consider them. If you look at, so there's a traditional like the Pareto distribution or power law of pond distribution, right? And we found that with our data. There's a little bit tapering off for very small ones. I don't know whether it's an artifact or it's a real. So that's something else. But if you look at area, they actually have a really large area footprint, and they can be hotspots for a lot of biogeochemical activities. And then we compare our data, so this OptiStar POM algorithm, with some of the reference polygons we manually check, and the NHD water body, the national data set available to quantify those. We found that is our method identify more water bodies than the national, the NHD data set, which mostly based on elevation, DEM modeling. But some of them might be vernal or ephemeral water bodies, so we're still working on mapping the permanence of those water bodies as well. So these are like little things we're going on, but with that I want to end and thank many like funders and collaborators and the amazing team I have at Cornell. And thanks everyone. A little bit over time, am I over time?
Good. Thank you very much, Dr. Xu, for a wonderful talk, and we'll now take some questions.
Herbert.
Thanks for your talk. For the Cape, so I should say I work with Evan.
Okay. Okay.
For the Cape, I found it interesting that you saw that, yes, with the increased Cape values, you get higher mortality. Did your group look at all about if Cape is increasing over time? Because at least in some of the papers we've looked at, there are different results from that.
Yeah.
So I'll repeat the question.
Okay, sure.
So are the Cape values increasing over time?
It's a very good question, and I actually discussed with Ivan about it. There are papers analyzing the trend, right? But there are also atmospheric science people saying that it's uncertain and untrustworthy given the reanalysis data we're doing. So that's why I'm saying it's still in progress. We're not sure. If you know a good way to quantify that, I'm happy to check that. But that's a very good question. I don't think I have an answer for that. Yes.
You talked about the changes in the woody growth efficiency of the allocation. I guess you were linking that to changes in allocation maybe to below ground root growth or exudation, something like that.
Yep.
And I was trying to think through like what other changes might be going on in those forests that might be driving some of those allocation changes. Have you thought about that at all? Like why would the trees be?
Yep. So we have some alternative hypothesis that's, at least for now, we don't have data to test. First is allometry change. The trees, the diameter height relationship or branch pattern is different because we only have basal diameter change, right? So maybe they're just having different shapes in the crown. That's one thing. And there's some evidence saying that the under higher CO2, the trees become slimmer. So that means although your diameter change doesn't change, your per diameter height change is increasing. That means that you still have more carbon accumulation, right? But again, most of those are based on gymnosperms and in temperate ecosystems. I haven't seen any reports in the tropical forests. That's one big hypothesis. The other is increased canopy turnover, like the short-term tissues like leaves production and other things. But I feel like you have to really crank it up to explain that lack of growth signal. And all the others currently I don't know. We talk about VOC or other potential pathways, but the magnitude is just different. So our best bet is still something below ground because one of the mature forest phase experiment in Australia shows that they found after 3 years of elevated CO2, they found increase in GPP, no change in NPP, increase in soil respiration. So their explanation is that either because of increased fine root turnover or priming, that really goes into soil respiration. So that's the only ecosystem level evidence we have right now. So I'm really hoping like Amazon phase is going to start this year, and so we can get some in situ measurement to see what's going on. Does the soil respiration really go up? And I put my bets in soil below ground and soil respiration is because during the drought, like 2000, the ENSO, the soil respiration also have a big signal. I feel like the plants downregulate soil respiration. Oh, well, I wouldn't say downregulate soil respiration. Soil respiration was changed by a lot during the drought. So that suggests something's going on. The plants might be able to regulate it very quickly, and then they bounce back after that. So that's partly why I put my bets on that. Yeah. Yes, Winslow.
Super cool talk. Thank you. I was really excited about the optical remote sensing algorithm for tracking mortality. And I had two questions related to that. The first is, what is the potential for using that in other forest biomes? So could you extend it globally? And then the second question is, are you thinking about using that data set as something that can be assimilated or benchmarking your model with to be able to constrain those elements in our system?
Yeah, exactly. It's a very good question. So this approach has been applied, the method originated and was tested in California during the chronic California drought. And the original idea is to use that as early warning signal. And we found we can't do that in the tropics. So the early warning signal they found in California because of the chronic stress. So you can see that rank up in classical dynamic system literature is increasing autocorrelation. We can't find that in the acute drought, in the tropics. But what we found is, in fact, it can use to predict the magnitude. So I think it's very likely to be working, and the algorithm should be straightforward. The only thing I feel like is to be really careful to remove seasonality or seasonal changes and really extract the high frequency variability. Otherwise, the autocorrelation is not trustworthy. Yeah. And then for the second question, so for sure, I think that's the goal of creating that data set. So create a benchmark, spatially explicit benchmark for the big models to tune their parameters. And for me, for our process-based model, I'm more interested in the kind of the sensitivity part, because we're not just trying to tune that parameter right. We want to know what process do we need to add, right? We need to create a function to describe the sensitivity to CAPE. We need a function to constrain our sensitivity to temperature and other things. So those, instead of the pattern, but the sensitivity is what I feel more excited about. But the patterns for sure would be very important for the big models. Yeah.
All right, I think we'll stop there. It's noon, so thanks again, Dr. Xu, and we'll see people in person and the lunchroom for lunch. So thank you.
Thank you.